We're now moving on to a new session, Matsu Salmon in a Warming World, and Dan Rinella is going to start us off talking about Deshka River temperature monitoring and salmon studies.
Title of Rinella. And Becky, do we have a laser pointer here?
Yes.
Is it this thing? What button do I use? Oh, there we go. There we go. Oh, that's advancing. Okay, okay. Hi, everybody. Yeah, I've missed the symposium for the last couple of years, so I think it's time for an update on all the work we've been doing related to warm water in the Deshka River and salmon. My name's Dan Rinella. I'm a fisheries biologist with the U.S. Fish and Wildlife Service in the Anchorage field office. And we've been working on the Deshka for quite a few years now. This is kind of a more or less a high-level overview talk of all the things we have going on. There's just too many things to really dig into any one of them in any detail. I'd like to start out with some acknowledgments. First off, John Gerken, my branch chief in the back here, he supported this work in every way that a boss possibly could. Sue Mauger with Cook Inletkeeper really helped us get the ball rolling on this and got us squared away with all the temperature monitoring stuff, and did a bunch of canoe trips with us, getting the network set up, and just got us off on the right foot. Anna Marie Benson with Fish and Wildlife Service is leading the analysis and the modeling. The Palmer ADF&G office has helped a bunch with the radio tagging at their weir and some of the juvenile sampling that we've been doing. We've had a whole army of biologists and technicians and interns who've helped with in the field and in the lab and then with data cleaning and management and that sort of thing too. And then Fish and Wildlife Service Water Resources, in particular Meg Purdue, is archiving our millions and millions of temperature data points for us. And then in terms of funding, Matsu Partnership gave us the initial funding for this work, and we kind of got the ball rolling, and the conversation started here as part of the partnership. ADF&G and Cook Inletkeeper helped in kind in the field, and then finally U.S. Fish and Wildlife Service provided a lot of the funding, including several grants through the Science Support Partnership that have led to student support for graduate students at UAF who've been involved with the project. So why are we focused on the Deshka? It is a historically productive Chinook and Coho salmon producer. Historically, it's been the largest producer of king salmon in the northern Cook Inlet, and as we've heard from a number of speakers, that salmon populations in the valley are not doing well right now. That's a main part of our interest, and we're interested in it because it gets warm. Its warmth has probably historically led to its productivity, but we believe that in a lot of years it just becomes too warm to really function as salmon habitat, and we'll get into that a little bit down the road here. Another reason that we're interested in it is because it is logistically simple to work there, and it's road accessible. And it's this flat and low-lying area here in the Matsu Valley, and I think that the lessons we learn here can apply to all this kind of flat and lowland area out in the southwestern part of Alaska, where a lot of salmon runs are also in decline right now. And also there's a lot of interagency interest aside from just the partnership. Climate change has been a priority in the Fish and Wildlife Service's regional strategic intent. This type of work has been highlighted in the Freshwater Temperature Action Plan, and then with the possible ESA listing for Gulf of Alaska Chinook salmon, there's going to be a lot more interest in studying Chinook in the near future. This figure's been shown at this meeting a number of times over the years. The point here is that this is from Sue Mauger's work, where she monitored water temperature in the main stem of 50 different Cook Inlet salmon streams. The point here is that it's these warm streams up here. The open circles represent the maximum weekly maximum temperature in 2009. The bars extending to the right represent projections of that statistic in the 2030s and then in the 2060s. So up here in the upper right-hand corner, we have all these lowland streams that are already warm. They're already exceeding all of these EPA thermal criteria for salmon. And the bars extend furthest to the right, so that means we're expecting them to warm the most. And then we also have some evidence that warm water correlates with low Chinook salmon productivity in the Deshka already. On the left, the Deshka data points are on the red. Each one represents one brood year. The trend line is also on the red. The left panel here, the take home, is that for runs that spawn during warm years, fewer of their young come back in subsequent years. And on the right panel, the take home here is that for juvenile Chinook salmon that rear during warm years, fewer of them come back as adults in subsequent years. So onto the actual Deshka work. There's two core components to this work. One is a temperature network across the watershed, and the second component is our juvenile salmon surveys, and these two components work together in a lot of ways. And then we've added a lot of projects over the years that capitalize on one or both of these components, and those have led to a lot of fruitful collaborations. So first of all, the temperature network. Objectives here were to build a watershed scale model to characterize current, past, and future thermal conditions across the entire watershed. And in order to build this model, we needed a lot of temperature data at the right spatial scales. And then another objective is to provide site-specific temperature data for fish studies. And I'll get into that a little bit more later. So method-wise, we log temperature year-round at 82 sites across the watershed. That's all of these dots right here. We've been doing that since 2017. 23 million temperature data points. We have 2 loggers at each site, so it's actually 46 million temperature data points that we currently have. They're mainly focused on tributary junctions. And so access-wise, you can see this string of sites right here is Petersville Road. This string of sites right here is Oil Well Road. So some of the sites are road accessible. And then the rest we access by launching a canoe at one of these 2 roads and then just floating down all the way to the mouth for a pickup at the mouth. And like just off the top of my head, we've canoed somewhere between 2,000 and 3,000 miles collectively up to this point, managing this temperature network and sampling salmon. Anna Marie Benson has worked on a Bayesian spatial stream network model. We have a great temperature model. It takes 24 hours to run on the UAF supercomputer. So when we do a simulation, we tend to choose it carefully. And the point here is that the main environmental inputs to the model are air, temperature, and recent precipitation. So if we have those inputs from past, like from the Talkeetna Airport, we can reconstruct or hindcast previous temperature conditions across the watershed. And if we use downscaled climate projections, we can forecast into the future across the watershed. And then the juvenile salmon surveys. Goals here are to estimate how temperature influences the growth rates and the distribution and relative abundance of juvenile salmon, and then to forecast changes in the abundance and distribution of thermally suitable habitats for different life stages. And we're especially interested in spawning and rearing habitat. And then also we're interested in determining if juvenile abundance can help forecast the size of future salmon runs. So like, is the number of Chinook that we're sampling this year reflective of how many of those fish come back after they go to sea and mature? Methods-wise, we're sampling juvenile Chinook and Coho across the growing season at all of the same 82 sites where we're logging temperature. We started that in 2019. We're probably done with that this year. We sampled in excess of 10,000 fish. It's 10 minnow traps per site, a 1-hour soak. We measure length and weight. And then from a subset, we take otoliths for some age studies, some growth studies, and then we take stomachs and body composition from those fish as well. And so for the rest of the presentation, I'm just gonna, this is a really complicated project with a lot of collaborations and moving parts. So I'm just gonna focus on a few questions that we're working on answering kind of right now and that we've made some progress on. These questions are, what is the distribution of thermally suitable habitat in a normal year versus a warm year? For the warm year, we're focused on 2019, which was a record heat wave. And we expect that to become sort of the new norm in future decades. So it's sort of a little glimpse into the future of this watershed. And we're interested in the future of rearing habitat, which we define as temperature less than 18 degrees, and then spawning and incubating habitat, which we define as temperatures less than or equal to 13 degrees, and these are EPA's water quality criteria. Second question, are juvenile salmon showing signs of heat stress, physiological heat stress? Is warm water impacting juvenile salmon growth? How are Chinook spawners coping with warm water, and then finally, how many juvenile salmon are pike eating, and will it get worse? So moving on, so this is some of our model output, and this is really, really basic stuff. We ran I think 2,000 iterations instead of the standard 1,000 iterations, so we get the model to run like in a day instead of 14 days. So here's the warm year, right? And we're spanning from June to August. Yellow corresponds with a high probability, a probability of 1 of exceeding 18 degrees. And you can see that for about a month during that warm year, almost the entire watershed was too warm for salmon rearing. And then even on into August, most of the watershed was too warm for salmon rearing. And then compare that to a cold year. And you can see a few little yellow dots here in July. But for the most part, the whole watershed was thermally suitable during that time. Here's the same type of map, and now we're looking at 13 degrees for spawning habitat. And this is the kind of startling thing, is that even in the normal year, the entire watershed is according to EPA's criterion, is too warm for salmon spawning. Second question, are juvenile salmon showing signs of heat stress? These plots are from a collaboration with Vanessa Von Biela at USGS. The first thing we did some lab studies with hatchery juvenile Chinook and Coho to see at what temperature they start showing signs of physiological heat stress. Vanessa's presented at this meeting before. For Chinook salmon, it happens at 21 degrees. For Coho, it happens at 23 degrees. So here's some data from wild Chinook in the main stem Deshka. We see at about 18 degrees, half of them are showing signs of physiological heat stress. This is at HSP 70. By the time we hit 22 degrees at the sampling site, 100% of them are showing signs of heat stress. And then in terms of juvenile salmon growth, hang tight. John Hermus is presenting next. This is his thesis project at UAF. He'll fill us in on all this stuff. And then how are Chinook coping with warming water? Mike and Bailey have a poster in the back of the room. Hopefully you had a chance to check that out. I'd like to point out just one thing on this plot. Here's that 18-degree criterion for migration. And so this fish is experiencing, during the day, at these peaks, temperature's too warm for migration, but during night things cool down, and we see these fish mostly moving at night, so they seem to be kind of coping with it in that way. But when it comes to the kind of the core spawning period right here, the water, this fish was spawning in water that was physiological too warm for itself. So we put out almost 100 tags in the last 2 years. We've got almost 80 of them back. Our recovery rate has been great. We're interested in continuing this work. We are struggling to meet the non-federal match to buy more tags. So if anybody has some ideas for that, that would be hugely helpful. And then I'll wrap up with some pike work. This was Ben Rich's thesis through UAF. He relied on our water temperature network as part of his thesis work here. He noticed, compared to some older work that ADF and G did, pike are consuming fewer salmon now just because there aren't as many of them to consume. But by his estimates, they are currently consuming 2,000 to 2 million juvenile salmon a year. And then he did some modeling to show that their energy demand is going to increase by 6 to 12% as the water warms up. So we can expect them to continue to eat more salmon as things warm. Next step, we have some manuscripts coming out. Be on the lookout for those. We hope to continue the temperature network and some carcass surveys indefinitely to see if fish are actually spawning successfully or not. A couple graduate research projects to finish up. And then we're going to focus a little bit on this question, does juvenile salmon abundance now reflect does it predict to the number of adult salmon that come back subsequently? So I'm out of time. That's it. Thank you.
Do you have any questions for Dan?
Too much information.
Yeah. Okay, well, thanks, Dan. We'll queue up John.
Wait,
Okay.
Oh, sorry, go for it, yeah.
I just have one. We're seeing Coho and Chinook challenges all over these things.
Sure.
Why do you disseminate between temperature and when it's common in streams that are not having a temperature challenge? What else do you look at?
Well, they share a common environment at sea, and that's probably a huge part of the problem. Warm water at sea has changed the food web extensively, and the taxonomic composition of the prey has shifted to organisms that are less nutritious, and then and in warmer water they're storing less lipids. So there's a big problem with the marine side of things, and here we're focused on the freshwater side of things. But it's probably a 1-2 punch. Question in the back?
Yeah, how are the Dolly Gardens doing in the warmer water?
Boy, that's a good question. That's a good question. They're probably pretty sensitive to warm water, but it's not something that we're monitoring.
Does
Yeah.
the color of the water have anything to do with the temperature of water?
Boy, it sure does. It sure does. And then for the sake of time, I didn't go into that a little bit, but let me scroll back to this aerial view of the Deshka, and it really captures why it's so warm. And it's not alone. There's a lot of streams like this across the landscape, right? So that upper right-hand panel, the maximum elevation in the watershed is about 1,200 feet. It's very low. It doesn't have any glaciers, no persistent snowpack. So the snowpack's gone by like May. The stream channels are not shaded. A lot of people have been out there or on similar rivers, but the riparian zones are just grass. There's no trees that give any canopy cover to shade the stream. They're flat and slow and snaky, so the residence time is really high. The water just has a lot of time to warm. And that black tannic water absorbs a lot more radiation than clear water would too. So all those things sort of conspire to make it super warm. Yeah. And it's interesting. I'm in the literature on warm rivers a bit and reading papers from the lower 48, and they talk about warm water being above 20, and here we get warm water that's in the high 20s, and I think that the tannins have probably a lot to do with that, and our much longer days during the summer have a lot to do with that. And then our warm water also coincides with the exact period of time where Chinook salmon are migrating upriver and spawning too, so it's hitting them at the absolute worst possible time. Is that it?
Okay, thanks, Dan. Okay, our next speaker is John Hermus, and he'll be talking about the effects of heat stress on juvenile Chinook and Coho salmon growth in the Deshka.
All right. Is that okay? Cool. Well, hi. My name is John Hermus, and it's awesome to follow Dan after a great overview of the Deshka. And I'll get to give a little bit of specifics on one smaller portion of that. So I'm a master's student at UIF currently, but previously worked at the Anchorage field office with Dan and John Gherkin and a whole bunch of other folks as a biologist there for a few years, where I collected this data and then got to bring it to school. So we'll get right into it. So to start with a quick background of why the specifics of my research is important, after Dan gave that very good overall background. As we're aware, the climate's warming globally, but it's happening at a much faster rate at high latitudes, such as Alaska. And coinciding with this warming, like we've heard about through the symposium, Chinook salmon populations are declining rapidly throughout the state. And that's leading to fisheries closures in sport, subsistence, and commercial closures. And a lot of the drivers of this are still fairly unknown, outside of the fact that we think it's because of warming waters. So in recent years, along with this, the temperatures within our freshwater within the state have been warming and exceeding past the upper thermal limits of salmon. And for juvenile salmon, those upper thermal limits are around 21 degrees, where they're becoming stressed and having troubles with growth. And as we saw from this press release from the Cook Inletkeeper, the Deshka hit almost 28 degrees in 2019. So that led us to wonder, how are these freshwater conditions, these warming freshwater conditions, affecting the juvenile salmon and the Deshka? And we're really curious about the growth of them, or at least I was. So with that, we hypothesized that Alaska's warmest freshwater temperatures are already impairing the growth of juvenile salmon, and we figured that was due to the direct effects of heat stress or indirectly through metabolic rates that are exceeding consumption. So simply put, we just figured that the higher water temperature mean that they needed to consume more food to match those metabolic rates, and we didn't think that was becoming impossible within our river systems. And this is really important for something called, in the framework of the critical size, critical period hypothesis. And this hypothesis states that if salmon are entering the saltwater at a smaller size, they have a decreased chance of survival when compared to larger fish because they can't avoid gap-limited predators, and they also have just a lower chance of escaping predators. And if this is happening, along with our salmon having decreased growth due to this warming water, this could be contributing to the population declines we're seeing throughout our state. So Dan already gave a very nice Deshka overview, but just a quick touch on that. As we learned, it's a non-glacial river and is draining that whole wetland of the Western Sioux. So it's moving very slowly, and it's consistently above 20 degrees in the summer months. And like we learned, this has historically had the largest Chinook salmon run in the northern Cook Inlet, and that run has plummeted. So we feel that this river is representative of salmon systems across the state that are also at this upper thermal limit of salmon. And it can also be used to understand the future change in systems that are cooler that may be warming in the coming decades. So once we had these questions, we began collecting data, and as we learned, the Deshka has this great thermal monitoring system through it, so we decided to pair fish sampling with those thermal monitoring stations, and those stations are recording water temperature every 15 minutes annually, so we were able to pair our growth and heat stress metrics with water temperature, continuous water temperature data. And for my project, we paired sampling at 68 temperature monitoring locations throughout the watershed, and then we trapped with baited minnow traps, and we paired this with the overall trapping that was happening for the rest of the projects. But for my project, we collected a subset of fish to be processed for growth and heat stress. And for growth, we were looking at otolith daily increment widths, and for heat stress, we're looking at HSP 70, so heat shock protein 70, and I'm going to get into what those are next to have an understanding of how this works makes sense. And while we were out there trapping, we did trap fish in almost 26 degrees Celsius, so we were very successful in trapping fish over that thermal limit that we were hoping to trap over. So, to start at the very basics, if anyone's not familiar with an otolith, it's the inner ear bone of a fish, and you can see how it's circled on that diagram on the right. Those are the sagittal otoliths, the ones that I processed. And we were processing about 40 to 70 millimeter fish, so these otoliths are about the size of a large grain of sand, so it was pretty tedious work, but pretty rewarding when we get the pictures that I'll show in a second. So in salmon and other juvenile fish, they lay increments at a daily rate, which then allow us to correlate that to growth, because increment width has been correlated to somatic growth in juvenile salmon. And this is a final picture of what those otoliths look like once we complete them. So we polish them and then photograph them at 40x magnification, and then we use a program called Image Pro to process this data, where we mark every single increment and then measure the width between them. And then from that, we're able to calculate a 10-day average for each fish to use as a growth metric for this project. And that 10-day matches with the amount of time heat stress stays in the tissue of the fish that we detect next, which is the HSP 70. So heat shock protein 70 is a core component to cellular heat shock response, and it's used widely in fisheries as a bioindicator for heat stress. And what HSP 70 does is it's produced in the cells when warm temperatures begin to damage proteins that are required for normal cell function. And it's produced because it assists proteins in maintaining the proper folding and repair damage, as well as preventing aggregation of proteins in those cells. So simply, when a fish is stressed, those cells are having troubles, and HSP 70 comes along to make sure it can, to do its best to help it maintain that proper function so that fish can do well and not die for as long as possible. So we're looking at the pre-mortality side of the heat stress in this project. And then, so this figure is from Vanessa Von Beel's previous work, and we can see that in lab studies, Chinook are being stressed at around 21 degrees Celsius is when they're starting to really show increased levels of heat stress for these fish. And it's kind of just a fun side. The heat stress samples have to be frozen instantly, so we carried this really big nitrogen shipper around with us, then the Deshka for 4 whole years with all the other sampling gear. And this is a picture of the confluence of the Deshka. So that's Croto Creek coming in and then Moose Creek on the right. So kind of a special place on the Deshka. I thought it'd be nice to share. And I'm just getting into my results, and this past couple months, about a month now, I've been able to start digging into the data while I'm working on my master's, and excited to share some of the very beginnings of that. So I want to start off with the trends of water temperature we've been seeing over the 4 years, or we saw over the 4 years that we monitored the Deshka for this work. And it ended up working out really nicely for my project because I've been calculating, or working in the framework that we have 2 warm years and 2 cool years. And everyone can probably remember who was here that 2019 was a very abnormally warm year for Alaska. And we can see the dashed line is the 21 degrees Celsius where salmon are becoming stressed. And in 2019, the majority of the sites in the Deshka were over that metric for almost a month period of time. And then in 2020 and 2021, we did occasionally break over that on a few sites, but it wasn't for a long or extended period of time and not on all sites. And then 2022 again breached that 21 degrees Celsius for an extended period of time, although it wasn't quite as long as the 2019 heat wave. So lending us to kind of have an idea that maybe these are 2 potential future years in the Deshka and maybe some past or present years, and allowed us to look at kind of the cool effects and warm effects at an annual scale, which is what I'm going to share today. So if we take this a step further and then look at the heat stress results, this is a figure showing HSP 70, and we look at it in nanograms, HSP 70 to milligrams total protein. And for these wild fish, I'm seeing decreased growth at around 5 of these units. So that's where I'm suggesting those fish are becoming stressed. And we can see on the warm year of 2019, substantially more fish are stressed than the 2 cool years, as well as they have a lot higher, or present a lot higher rates of HSP in the tissue. And then the 2 cool years, we have substantially lower to almost no stressed fish in 2021. And then again in 2022, when it was a warmer year, but not quite as extreme as 2020 or 2019, there is again increased heat stress in those fish. So from this, we're getting an idea that extended temps above 21 degrees Celsius appear to be inducing heat stress in the juvenile Chinook of the Deshka River. And then, if we take that a little further to growth, we're seeing a continued similar pattern where after just running a quick ANOVA, we do get significance in these differences. So for the 2019, as we talked that was the very warm year, there's a significant difference of decreased growth for the Chinook with their otolith increment widths on the Deshka in that year compared to the 2 cool years. We can see as they're signified by the letters atop of the box plot, while the 2 cool years of 2020 and 2021 had a higher growth that stayed consistent between both of those years. And then the year of 2022, we did see decreased growth, although it wasn't significantly different from either 2019 or the cool years. So from this, we're getting an idea that extended heat waves, rising water temperatures above 21 degrees appear to be having negative effects on Chinook salmon growth in the Deshka River watershed. And that it was our hypothesis, although it is a little bit unfortunate to see that the river is now pushing the temperatures into ranges that are affecting the salmon in the fresh water while they are growing. And as I said, I'm just getting started with my analysis, so I have a lot more to do, so I'm excited to continue exploring the data and seeing other trends that will come out. And I plan to create a mixed effects model that includes water temperature, HSP, and then I didn't get to talk on it today because just of a time limited, but I have stomach fullness data because that's a very important metric when looking at growth. And then I also have total fish dry mass to show the lipid levels or how healthy those fish are. And then I'm going to hopefully add discharge year in sight to really get an idea of all the things affecting the growth of Chinook in this watershed. And then lastly, like we've heard, Coho also appear to be struggling in the recent years. So I have a data set for all of the same information for Coho. So I will also dive in and look at the effects of heat stress and growth on Coho throughout the watershed. So that's just a quick introduction of a portion of my thesis, and I'm really happy I got to share that today. And of course, I have to thank everyone at the U.S. Fish and Wildlife for my time there and all the field techs that helped. Specifically, Ben Rich was huge for 4 years, and Tim Erickson helped process about half or more of the otoliths that we did, and those took winters, so that was a huge help. And then, of course, my committee and my co-authors, Jeff, my advisor, and Dan, Vanessa Von Beal at U.S. Fish and Wildlife, and Jeff Felke. So thanks a lot, and happy to take any questions.
Any questions?
Yeah, I like that talk. I was just curious if you had any other kind of response measures you were looking at besides heat shock protein. I've seen a lot of work recently looking at these gene transcription factors, and and kind of doing more of a non-targeted assessment of what genes maybe turn it on or off.
Yeah. So Vanessa has done that in that lab study I shared. Her paper looked into a large assortment of other genes and proteins, and through that she ended up choosing the selected to move forward with HSP as a viable one for field work. So in the lab that's been tested, and HSP is the one that's been applied to the field work currently.
John,
Cool.
I have a quick question. Could you take the growth from 2019 and try to project if you thought when they smolted, they're so small that you think they would have trouble getting through their first year in the ocean?
Yeah, I don't have any idea of that yet. And I've just been working specifically on the growth, so I don't know those requirements of what sizes are needed to have success. I think that seeing a significant decrease in growth, I think that it could most definitely have an effect on that ocean success. And then also just having a bunch of other physiological stressors while in fresh water could definitely be decreasing survival for other reasons as well. Oh.
I can just shout.
Okay.
Really cool. I'm curious, and maybe this is analysis that you have planned for the future, but when you break out your data set by like tributary or trapping location, are you seeing like interesting correlations between like worm tribs and growth or core tribs and growth?
Yeah, definitely.
Like, yeah, with the site-specific growth.
Yeah, I'm just getting into that, and there appears to be no site-specific difference, although the temperature difference is very large. So, like the site-to-site, I guess it would be probably more representing like the habitat at that site. Doesn't appear to be affecting it, at least on the Deshka. But there is, the temperature at each of those sites is appearing to continuously be a significant influence on the growth of those fish and the heat stress. So does that answer fairly? Okay, yeah. Cool.
Thank you, John.
Thank you.
Okay, next up we have Erin Larson, and she'll be talking about juvenile salmon distribution and body condition, the influence of thermal regimes in the Little Susitna.
Awesome, thanks. I'm going to take you a little further east, and we're going to think about the Little Susitna, but thanks to Dan and John for setting up sort of the why we should care about juvenile salmon and temperature, and some of the work that they're doing there that we're sort of mirroring over in the Little Sioux, just a few years removed. So just to run folks through a brief history of the work on the Little Susitna, this was started in part by Becky Shaftel and Dan Rinella, and I've inherited this project. So a temperature monitoring network was set up at 28 sites, so 13 in the main stem and 15 on tributaries in late fall 2019. And then we started doing some juvenile salmon monitoring, really mirroring the work on the Deshka at all sites in the early summer of 2022. And so today I'll be talking about, we now have 4 full years of temperature data, 3 full years or seasons of juvenile salmon sampling in both the early and late seasons to look at growth throughout the season at a population level. And then moving forward, we'll be continuing this temperature monitoring to capture at least 5 full years of data. And so I know folks in this room are probably very familiar with the Little Susitna drainage, but again, we're just moving a little further east and up into the mountains, up into the Talkeetnas. And again, these sites are paired main stem tributary sites. So in pink here are the main stem sites, and in this gray or blue color are the tributary temperature sites. And so our sites range up from Archangel Creek and some main stem sites just above it, up at Hatcher's Pass, down to the boat launch and campground off KGB Road. And so in terms of temperature methods, I feel like this room is probably very familiar with various ways of measuring temperature. We have paired temperature loggers at each site, and those paired tributary and main stem sites. We maintain and download loggers in the early summer and fall to make sure that we're able to capture the full summer, and that way if we lose loggers during the winter, we've gotten the data during the prior year. And then we QC all our data and upload to AKTemp. So all the temperature data I'll be sharing today is all publicly available on the AKTemp database. In terms of fish methods, again, we're mirroring the work on the Deshka, so we have 10 minnow traps that soak for an hour at each site. We've played around a little bit with longer soak times, deploying more traps, because we've had issues in some years with actually capturing enough fish, unfortunately, and that hasn't really seemed to make much of a difference, so we've sort of stuck with that 10 traps for an hour. We record all fish that we capture, and then weigh and measure any of the salmon species that we collect. And then we send 5 vouchers from each site for each of the Coho and Chinook species to Fish and Wildlife Service from each sampling time point. And so just to set up a little bit of context for comparing the Deshka and Little Susitna, it's probably not going to be surprising to anyone after the last few talks that the Deshka is very warm. So this is, these are figures looking at historical data that's been collected, temperature data on the Deshka, and I just want to walk you through these thermal regime categories. I'm going to walk away and shout or talk loudly. So we have cold stable thermal regimes, cold variable, and then warm long and warm variable. Unsurprisingly, the Deshka mostly falls into these warm categories, both on the main stem and in the tributaries during most years. In comparison, on the Little Susitna, we mostly see the cold variable, but we also do see some sites in some years where we see more representation of the warm long or cold stable categories. So there is thermal heterogeneity both within the Little Susitna drainage, but then also across years as well. And if you're looking at the 2024 data and wondering what happened, when we went back to sample downstream of the Parks Highway, the water was really high, and so Dustin and Mike weren't really actually able to access some of our loggers to download them at the end of this summer. So we'll be going back and getting that data next summer. And so when we sort of zoom out and aggregate that information, again, the Deshka, unsurprisingly, is very warm. The Little Sioux is cold, but there is that interannual variation as well. And that sort of highlights the importance of doing these types of temperature monitoring networks across multiple years, which I know Sue Mauger has really advocated for in a lot of her work as well. And so when we start to layer this temperature data onto thinking about its effects on fish, this data, I'll walk you through this here, is all temperature data that we've collected at the Little Susitna Weir, overlaid with the run counts from Fish and Game at the Weir. And so when we think about the timing of when Chinook are migrating up the Little Susitna River, we have fish counts on our left axis and then temperature on the right axis. That red line is an 18-degree threshold, and that 20-degree threshold is shown right above in black. And so you can see that on the whole, the Little Sioux is cold, unsurprisingly. And so we actually haven't seen from the 2016 was a previous year where some temperature data had been collected, but 2020 to 2024, we don't really see concerningly warm temperatures at the Weir. If we sort of shift forward in the summer, or shift later in the summer, to when we'd see Coho migrating past the Weir. Similarly, mostly cool temperatures at the Weir, so not evidence of temperatures that could cause heat stress for migrating Coho. If we think about juvenile growth across our different sites, this is a noisy figure, so I'll walk you through it. It's not showing up super well on the projector, but there's sort of a pink band in that optimal growth range for juvenile growth. And our sites are laid out in terms of their distance from Cook Inlet, so lower numbers are further down in the river system, higher numbers are further up in the watershed. And because the Little Susitna is such a cold drainage, we mostly see that those lower down in the drainage sites have more suitable temperature growth habitat, suitable temperatures for juvenile growth, and that those upstream sites are a little on the cooler side. So as we sort of think towards the future, we might be seeing a shift in where there is more thermally optimal habitat for juvenile growth in the watershed, higher up in the watershed. As I alluded to before, we did have some challenges with trapping efforts. So this figure here, I count on the y-axis and then the proportion of successful trapping efforts at a given site throughout the 3 years that we've been doing juvenile sampling. So even if a site was a site where we have previously found juvenile fish, it didn't sort of guarantee that we would find fish there in a future year. And so some of our work was definitely challenged by not really being able to get a very large sample size of juvenile fish at a lot of our sites. Digging a little bit into the fish data, this is going to be data from 22, 23, and 2024. We've caught three species of salmon, Chinook, Coho, and sockeye, both early and late. In 2022, we didn't catch any sockeye. We caught just a few Chinook and a few Coho, and fork length is here on the bottom, so no huge differences between the early and late season. In 2023, we caught far more fish earlier in the season. We were challenged by really high flows later in the season for trapping, and actually weren't able to do the float portion of our study that year. And then this past summer, we caught kind of a wider range of sizes in both the early and late season. Sort of overlaying those, you can see that our just abundances have also were a lot lower this year than they were in 2022. And this is that same data, but just looking at densities instead of raw counts. So again, we're seeing some differences between years. I'm still also digging into this data since we just finished our last year of data collection this summer. I'm sort of looking at those site-specific differences. But interestingly, we don't see a very bimodal distribution in our Coho salmon, so not seeing like an evidence of age zeros and age ones, or we still need to sort of tease the data apart to see those two cohorts in this juvenile data. Getting into some body condition data, so this is almost like fish BMI, taking their weight divided by fork length. Across all of our sites, early in the season and late in the season, there's not huge differences between sampling years. But once we drill into some of the data at a site level, this is Coho Creek, which is 73 miles upriver from Cook Inlet. And there's possibly some differences between years, but the data is also pretty noisy. If we go further downstream to a tributary of Nancy Lakes, we see some big differences in body condition in early season versus later in the season. And then a tributary at River Mile 33, we see some differences in between years. And again, this is still pretty new data, so I'm still digging into some of it and trying to figure out what exactly it all means. In terms of future directions, we have one more year of funding through the Fish Habitat Partnership to do this work. We'll be going out next year to download and maintain our logger network, both in early summer and in late fall. We've been sending subsets of our fish, those 5 vouchers, to Fish and Wildlife Service to look at some other body condition metrics like those that John mentioned earlier, so excited to dig into some of that data as well, and to do some work with Dan and his group on doing some more in-depth comparisons of the Deshka and the Little Sioux. And so with that, I'd like to thank all the folks that have helped us out with fieldwork. It's not quite as fieldwork intensive as the Deshka work. There's a lot more road access really, but Fish and Wildlife Service has done a lot to support this work, especially Mike Johnson this summer was really crucial in helping us with some boat access. Molly Legg, Audrey Giddings, Preston Blumson, Brian Demento, and Noel Spencer all helped out with fieldwork as well. Thanks to the Fish Habitat Partnership for funding. And I just wanted to reiterate too that all temperature data from the Little Sioux work is available on AK Temp if you'd like to dig into it yourself or if you're curious to access it. With that, I'll take any questions.
Any questions for Erin?
I know it's dark. I feel like we can also turn on the lights if everyone needs some like energy boost. Oh, just my legs. I get lots of energy. Okay,
Okay,
great.
thank you, Erin. Okay, for our last talk about Matsu salmon in a warming world, Sue's going to tell us about the next water temperature action plan, which is building on a decade of collaboration and strategic actions.
All right, good afternoon. Thank you for sticking around. I can see you people leaving. So in some ways, we just heard kind of the punchline of like, what do you get when you collaborate for decades and have real strategic mindset? And we just saw an amazing array of research that is beyond my wildest dreams for where I thought we would be at this point in time when I was writing the first stream temperature action plan back in 2012. So I think it was a report I had promised for an aqua grant that Laura Eldred was no doubt my project officer for. And the goal was to try to put a plan together that was statewide, that wasn't anything I was committing to, but that I thought should happen without naming names of who should do the work or where the funding was going to come from. Like, what were my big dreams for temperature work in the next 10 or so years? And ultimately, I didn't dream big enough because we've actually accomplished an amazing amount of that work. And so I want to review some of the highlights of that and kind of get you thinking about, well, what are the next big goals in the next 10 years? You know, just imagine how much more we can do now that we actually have data. So the ultimate goal, the purpose of it, was to identify the highest priority actions for the next 5 to 10 years that will lead to greater protection of Alaska's salmon habitat as thermal change continues. And to me, the crux of it was that it had to be accomplished through collaboration and coordinated conversations. And that is really what the partnerships are about across the state. And so for me, the Matsu Symposium the symposium and the partnership have been a huge vehicle to get so much of this work done. So we had 5 goals that we identified, and this was with lots of conversation with lots of people around the state. And at that point, there was very little data, and we really wanted to understand what the current thermal regimes were for Alaska salmon streams across regions and the state, to help refine the data collection so that it actually had value to fishery managers and land managers and the ability to model. We had a little bit of inkling about stuff going on in the Deshka, and so really starting to think about, okay, how do we protect cold water anywhere we can find it? Because we think that we're going to have a problem in the future. There was some grandiose stuff about filling stream network data gaps, which is basically imagining having one stream layer for the entire state. That turned out to be wishing too much, and that is, another story. And then finally, to direct relevant fisheries and habitat research, which is what we've been listening to the last 2 days and certainly for the last 3 talks. So how are we doing and what still needs to be done? So this is the 2024 progress report. It will be written up here in the next month. And this also was put together both retrospectively and looking forward through more conversations. We had a big workshop with about 40 folks, some of whom are in this room, back in February of 24, bringing folks together who had been engaged in temperature work across the state to sort of assess where we're at and to help everybody put their big strategic thinking caps on. So I'm going to go through a few of the to me, the highlights of it feels so long ago to go back to 2012 and where we were and how far we've come. But one of the first things we wanted to do is just really describe those thermal regimes. What does it look like out there in the field? And so that required that we were going to monitor temperatures across all these environmental gradients that would help us understand regional diversity. And the Matsu was a huge part of that. The first real network that happened in Alaska was in Cook Inlet, and 20 sites were in the Matsu, and we had partnership funding from both the Kenai and the Matsu partnership to do this work. So 5 years of data collection, found lots of warm streams, some cold streams, and lots in the middle. So ultimately, our progress has been that we've collected data, we have published regional analyses for the whole Cook Inlet and for the Matsu. We have regional networks that are ongoing in Bristol Bay, Kodiak Archipelago, Southeast Alaska, and Copper River. And Bristol Bay, we're working on the manuscript for that regional assessment, and that should be out next year. So the work is continuing, and this idea of looking at a big region and getting a whole network of partners together to help collect data across that region has turned out to be a really successful model for this work. We need to continue to make sure we get those regional analyses done, but one of the things that we realized looking at AK OATS, which was at the time, the standard, it's a metadata base that tells us where are people actually collecting temperature, which in 2012 we had no idea. This came online in 2014. So the metadata base could tell us where our big data gaps are, and the Yukon and Kuskokwim are definitely regions of the state that have not had proper attention, and that we know from the last few years have seen some real warm temperatures and a lot of concern about those stocks. So we've helped do a big training of folks in the Kuskokwim and Yukon region to actually accomplish that. So those networks have just gotten started. So the other thing we realized back in 2012 was that although it seems these little temperature loggers, it's not like really hard technology to work with, but you can do any easy thing badly. And so collecting temperature, although in terms of water quality parameters is kind of low-hanging fruit, but if you don't actually give people the recipe book, they're going to come up with all kinds of ways to do it, and they're going to create data that doesn't really become comparable. And so what our goal was to try to put together a little cookbook for how you actually collect temperature data, what are the important things that you need to do, the standards that you need to meet to make your data worth sharing with your colleagues. At the time, there were some national ones. I had done like a little bit of effort for the network I was working with, but there wasn't anything really statewide. And so that was something that in the next couple of years really blossomed. We did publish a minimum data standards paper that is very concise. It really tells you what you need to know. We have longer versions of that talk a lot more about deployment, and the first starts at quality assurance. Meanwhile, the National Park Service and USGS also put out some new guidance for temperature collection. So that was a huge jump, right? You can encourage people to do something, but if you don't actually tell them how to do it, you can't expect too much. So those are all wonderful resources that are available for anybody who wants to collect information. One of the things that came up very clearly in our 2024 February conversation was that one of the biggest tricks is deployment methods. And I had started this work in non-glacial streams, mostly wadable, and a lot of the systems that we now need to know a lot more about are these huge, the Kuskokwim River. Where do you put a temperature logger in that river that you'll ever find again, or that is in any way representative of a broader temperature profile? And so that's really the kind of work that we need to do now, is to get a bunch of folks together and to talk about what has, probably most likely what hasn't worked, because we have a lot of lost loggers. But so, how do we come up with some new ideas for getting into some of these bigger systems and glacial systems that really, oh gosh, I gotta go much faster. So like here's the Susitna River, right? How do you put a temperature logger in there and hope to ever get it back? And then back in 2012, we didn't have a lot of examples of really good web mappers. That was sort of young technology at the time. And so we were looking at work that was going down in the Pacific Northwest. And we ultimately wanted to at least know where people were collecting data, so identifying sites. And so with the wizardry of Marcus Geist and his ability and desire to work with a lot of scientists who don't really know what we're asking him to do, he created AK Oats. And this was just like, who is collecting temperature where, what kind of methods are they using, and here's a phone number, give them a call if you want the data. And I didn't honestly think we were going to get beyond that. And so, again, I didn't dream big enough, because we now have AK Temp, which we talked about a bunch yesterday. It's been mentioned in the last 3 talks as like a place where this data exists now. So you can actually download all the data. We have, I don't have the stat with me. I think Marcus had it. There's like 23 different provider entities. We've got, I don't know, 68 million, 16 million. We have millions of data sets in there, and it is all has some metadata in there. You can graph it. You can really begin to understand what is out there. You can download it all, and there's some amazing videos that allow you to upload all, that help tell you how to upload your data. So you too can contribute to this really wonderful statewide resource. I'm very proud to see this happen. There was a long way to go. Another thing that we really wanted to do is to try to refine data collection in a way so that we weren't just collecting data everywhere, but we were being strategic about it and thinking about where do we need long-term data sets and how can it be useful for climate modeling or fisheries applications. And this is kind of still something that's ongoing in conversation, but I would say one of the huge successes is just all that work that's come out of the Deshka and our ability to actually model that system as well as the Little Susitna. There's a similar network built on the Gulcana, so we will have that information at some point. We also have many, many sites that now have 10 to 20-year data sets. And so that's actually giving us some real, we're creating a story now. We really understand how things change at a decadal scale. So exciting. It was clear from the conversations we had back in February that we need a lot more work in trying to get the people who might want this data, those decision makers, and the people, I'm the next speaker, so I can go long. But that we need to get the modelers, the fisheries managers in the same room and say, okay, so we have all this data and we want to talk to you about what you still need. Like, what are the things, where are the gaps for you, not just spatially, but like, what do you need? Do you need 5-year data sets, or is 1 year enough? Do we need coverage at 82 per a Deshka watershed size, or can we do something else? And so there's a lot of conversation to move forward to really understand and make sure that ultimately, like, we are not the stream temperature partnership. We are ultimately about salmon habitat, and we care about salmon. And so how do we make sure that we're collecting data that has great value for the fisheries and land managers? We did do a little of this work years ago in Southeast Alaska and pulled together a big working group and came up with some management applications of temperature data, but it was sort of hand-wavy and like, this is what you could use for it. This is how you could use it for culvert assessment, or this is how it could be helpful for you to prioritize invasive species areas that you should do some remediation on. I think we've got enough data now, enough examples, so we can actually give specifics about how to use this data. Again, you've got to show people what it is you're trying to accomplish. So I'm hoping that that'll be something that'll really take bloom in the next couple of years to have those conversations. And then, finally, again, this is about fish, and this is about understanding how temperatures now and in the future are going to impact our fish resources. And so it's really just so heartening to hear of all the work going on. There's significant volumes of new literature that's talking about juvenile growth in Bristol Bay. There's all the intensive monitoring and modeling we just heard about, heat shock proteins. We're having, very sadly, the Chinook and chum and Coho declines. But we can begin to understand why those are happening because we now have the data in the freshwater anyways. Papers about retreating glaciers and new habitat availability and overwintering habitats. So there's a lot going on, and that is just very exciting. And I think overall, I think we did pretty darn well for our first action plan. And so we now are going to, we're in the process, we've been having regular meetings with a group of folks to launch the next stream temperature action plan. And actually, we're not calling it a stream temperature action plan, it's a water temperature action plan, because we want to make sure that we're getting the lake data in there as well. And so to me, this is a great, cheerleading about the value of collaborating and having conversations with folks sort of ad nauseum. Andy, I don't know if it was you or Howard or Larry that used to make bets about how many times I was going to say temperature in a talk. I have been talking temperature for a very long time, and I guess I'd encourage you, and I guess maybe I'll sort of take my hat. Well, I guess I have to take questions. I guess I'll pause there before I go on to my next series of random thoughts. Does anybody have any questions? Yes, Mayor.
We were in D.C. for the national meeting. NASA put on a presentation. Apparently someone's cutting into, there's a private sector firm cutting into their space travel. So they're looking for other business lines. But apparently, is their data viable on temperature data collection for habitat?
Remote sensing, you mean? Yeah. Yeah, so, and I didn't do that part of this because I was going to run out of time. But yeah, we've been doing thermal imagery in a number of systems. That is currently very intensive work that's done with an overflight of a plane over a river system. But I suspect by the time the next action plan is being reviewed, we will be able to pull from satellites imagery or data from satellites to give us not just air temperature, but water and land temperatures. And I imagine it's probably farther along than I realize. But that will, I think, be a huge technological advance in this work, is we do not necessarily have to go to every stream and put a dear little data logger in there. So, yeah, does that answer your question? Okay. Anybody else? All right. Well, Jessica gave me the chance to do last words, and I don't have any idea what time it is at this point. 4? 30. Oh gosh. Okay, those will be faster than I thought. So this is likely. It won't be my last Matsu Salmon Symposium, but it'll be maybe take a break for a little bit. And I've been reflecting on what I have gotten out of these meetings, and thinking back to just yesterday morning and some of the inspiring words that were said about, we're not going to win this battle to save our fish by just hope and wishful thinking. That this is going to be something that requires a real choice and strategy. And I hope this conversation about the action plan is an example of how you really can create change, sometimes more incremental than you want it to be, and sometimes wildly faster than you thought. But you have to be pretty persistent and dogged about your desire to get to the purpose. And for us, I think, it isn't just about having healthy habitat. It's obviously to have the fish thoroughly thriving in those habitats. But as we go through this, the next however many years of low salmon runs, it will become increasingly easy for decision makers to decide, we don't need to worry that much about the habitat because there's no fish to worry about. Well, that kind of thinking will ultimately end up with no fish for the long time, because when things change, when we have some shifts in the ocean conditions for some period of time and the fish can start to rebuild in some of those stocks, we have to have the habitat waiting for them to come back to. And I think, it's easy to imagine when fish are everywhere, the value of the habitat. And I hope you don't lose sight of the fact that you are doing this work for the 20 years, the 30, the 50, the 100 years when we want to continue to see Matsu salmon thriving. So I encourage you to be obnoxiously persistent in whatever component of habitat work that you do, and that it is ultimately about the connecting and the networking that happens at meetings like this, and that the value of your 2 days in your seat here and at the coffee table is more valuable than months that you can spend in your office. And I really encourage you to show up. I just think showing up, getting people to know you, making them comfortable to call you is really a gift that you're giving to this work. And so I encourage you to continue to prioritize coming to this meeting and to continue to show up in person, hopefully not forever on Zooms, but that your presence matters. You do vote and give the import of what you're doing by being in the room. And it's been lovely to have 2 days full of really engaged people here, and I am very thankful to have been a part of it for my tenure, and I hope that you will all also have a great 20 years ahead of you. So I do have a long list of some thank yous to do. I do want to thank first and foremost, Jessica Speed, who you all know how much she puts into this, but she is doggedly persistent. She doesn't let you off the hook. She will always follow up. And once she gets your text number, look out. And I always appreciate, I'm like, oh, there she is, good. She's like keeping me on track. So please give a round of thanks to Jessica. And then, of course, there's Rick, who has been here every year that I've been here. And can't say enough. You do the hardest of the work, is keeping us all to have something for people to look at rather than us. So thank you for your time and appreciate you. Thanks to the planning committee, the timers, the moderators, the presenters, the students, Turkey Red, all the pieces that make this a special time to come together every November. I know that there are evaluation forms, hard copies in the back, and that Jessica will no doubt be sending them out by email as well. Did you want me to say? They're on your tables. No excuses. That feedback is really helpful because, you get kind of like, oh, that was so great, and then you forget about all the things that didn't maybe go quite as you had hoped. So please provide some, honest, helpful feedback, and next year will be even better. I'm very thankful for the weather this year. A little chilly, but hopefully it'll be a safe drive home for everybody, so do be careful going home. And our last order of business is we have to put this place back together. We have to put chairs together, stacks of 5, tables, 10 per rack. The racks are probably in the back there. Please, take care, good luck, and hope the field work is great, and thank you very much.